Welcome again to our course on biodesign, strategies for innovation and design on bioengineering. Today we're going to take a look at the regulatory affairs basics module. So let's start. For today's class, the objectives are to understand the basic goals of the FDA and how this agency is organized, who the FDA is and what is there for. I want you to learn about the FDA medical device classification system and how this relates to the two main regulatory pathways for medical devices in the US, called the 510K and the PMA. Finally, I want you to develop a basic understanding of the requirements for regulatory approval within and outside the US. Nonetheless, you should know that in future modules, we're going to explore more in depth about the regulatory pathways that we're going to have really outside the US. Okay, so the FDA, which is the acronym for the Food and Drug Administration from the US, actually has a Center for Devices and Radiological Health called the CDRH. This CDRH is an office that constitutes a really large office with more than 1,000 dedicated, highly skilled, internationally renowned public health employees, including physicians, biologists, chemists, physics, engineers, statisticians, epidemiologists, microbiologists, nurses, doctors, pharmacologists, veterinarians, toxicologists, and many other specialists in public health, education, and communication. With all these highly skilled personnel, the CDRH and the FDA intends to create a context where regulatory approval for medical devices can be done in the right, appropriate way. The CDRH, as you're going to see, it has many different components, and actually there is an organizational chart of how this is constituted. First, for the CDRH, there is an office for the center director, and these branches out to many different offices which are going to be responsible for many of the determinations done on medical devices such like yours. First, there is the Office of Device Evaluation. Here is where all the medical devices, or most of them at least, are going to be determined. Then there is the Office of Science and Engineering Laboratories. This is where testing and testing experiment design is actually proved and analyzed throughout all the agency. There is an Office of Compliance which actually manages most of the post-market surveillance and compliance issues that the FDA surveils. There is the Office of Management Operations. There is the Office of In Vitro Diagnostic Devices, Evaluation and Safety. There is the Office of Surveillance of Biometrics and the Office of Communication and Education and Radiological Programs. As you're going to see, to know the organization of the CDRH gives you the opportunity, in the US at least, to know to which offices you have to refer yourself, and also to know that in collaboration with each other, these offices actually, at the end of the day, regulate your product and are going to be responsible for that. Okay, but going into more topic, what does the CDRH does within the FDA, this huge office in which all the devices, the medical devices, are going to be regulated? Well, they want to regulate medical devices and to eliminate unnecessary human exposure to man-made radiation from medical, occupational, and consumer products. This is the definition of the mission of the CDRH, but you should know also that this office is dedicated to providing enough evidence to show that the medical devices that are approved for the sale in the United States are actually as safe and effective as the manufacturers claim. In medical devices, the CDRH are going to be responsible for pursuing 5 different principal activities. First, they review all the cases of new medical devices for an application of submission for commercialization in the US. Then it sets also the standards and regulations for these medical devices, and some are gonna, you should think that might not change a lot, but with the advent of new medical technologies, biological devices, combination devices, and many others that are sometimes within the scope of drugs and devices and the combination of those, of course the creation of new standards and regulations is something critical for the success of this agency. Also, it monitors manufacturers' compliance with regulations and a quality system. It collects data on injuries or adverse events and the malfunctions of any medical device. Finally, it provides technical assistance to small manufacturers which may not have the capacity to actually cope with all the controls that a large transnational company could have to create new medical devices that are compelling for the medical system in the US. Something really interesting when you are talking about regulatory affairs is to compare what are the differences, at least regulatory, between devices and drugs. As you're going to see throughout the world, it's very common there are many officials and people that is providing services of regulatory affairs that has plenty of experience on drug regulation. Nonetheless, new device and development regulation, it's quite uncommon, at least outside the US. Nonetheless, it could be really common within the US and also a little bit of Europe. But at the end of the day, you're going to see that regulation of drugs is more common to be known. And so to know what are the key features that could be shared or not, it's critical for you to get into the idea of having a regulatory strategy on your biodesign process. So what we can see here is that there are different development features that either devices or drugs share. Nonetheless, the time or the money or the effort or even the way in which these development features are regulated or tested or at least thought at can be dramatically different. In this sense, for example, the rate of technological change in devices is much faster than drugs. And this is actually related to the amount of data and clinical data and research required to bring a new drug to the market. Even though a new device can come every single year for the same purpose with a little tweak or a little difference to have more competitive advantage in this space, a new drug is quite common to come. This is also a reason why many of the profits that drug and pharmaceutical companies want to earn are more representative of all this risk that they are coping through the technological development of these new drugs. But for this course, you might know, and you should know, that devices have a much faster rate of technological change than drugs. And thus, the way in which they should be approved and are approved is also faster. Also, the way in which bench testing is done and required, well, in medical devices, there is a medium to high amount of bench testing that it's done. In drugs, it's actually pretty low due to the absence of actually validated models for drug pharmacokinetic. In devices, as many have provide changes in anatomy physiology, that could also be tested on the bench. Which makes this a very valuable tool on device development, not as much as in drugs. Also, the value of clinical testing, well, it's pretty high in both. So you should know that clinical testing this in humans, which is the meaning of clinical, is as important as in devices as in drugs. In the case of reimbursement during clinical trials, which might be a characteristic very important when thinking about creating a new medical device, is actually very frequent on devices and rare on drugs. The influence of clinician technique on the results of the use of devices and drugs, in this case, there are dramatical differences that you are going to see between devices and drugs. In the case of devices, it's actually pretty high that the clinician's technique has an impact on the way outcomes go. Nonetheless, as many of the presentations of drugs are pills or some sort of delivery system that it's enteral, the technique in which these medications are given to patients are not as important as the molecule itself that it's going to act. So you're going to see that influence on this technique is very high on devices. The facility to assess performance after use is actually impressively higher in devices because of the way they are done, and also because of the same technological characteristics that makes trustability of medical devices higher than for drugs. Orphan classifications. Well, in the case of drugs, for a drug to be classified as an orphan drug or as a drug for an orphan disease, it has to have at least 200,000 patients throughout the world. In terms of medical devices, for a orphan disease device to be classified in such a way, you require only 4,000 patients. In regulatory classes, well, there is only one regulatory class in drugs. It is a drug, and that's it. But as you're going to see later on in this module, there are three basic types of devices in terms of medical devices, which we're going to see is determined by the risk associated to that device. And at the end, the full-scale studies required usually to regulate and approve a medical device is much less than a drug. In a standard way, in average, a device requires only one single full-scale study, while a drug requires at least two. Okay, so in general terms, what is the global concept on medical device regulation? Well, there are some basics which you might want to drill in your mind. You should know that for a medical device to get approved everywhere in the world, it should require a high degree of control on risk. Everywhere, the risk is one of the most important characteristics that you have to assess in order to have regulatory approval. It doesn't matter if it's Europe, if it's the US, Mexico, Latin America, Brazil, China, wherever you want to commercialize your product, to reduce risk as much as possible, or at least to show that the potential benefits of your technology outgo the potential risks of its use is one of the most important things to prove. With this in mind, consider that for the US, there is a very thorough analysis on the benefit and risk ratio in order to assess safety, which is a worldwide idea that must be met for a medical device to be regulated. But also in the specific case of the US and US harmonized nations, effectiveness is a component that is essential for device regulation. In this sense, throughout the world, generally, the standard should be to assess safety and efficacy or effectiveness of your products. Nonetheless, you should know that in some parts of the world, such as Europe, there is a much stronger focus on safety only than on safety and efficacy, which could be one of the reasons why many new medical products actually start being commercialized in Europe rather than in the US or other countries harmonized with this safety and efficacy system. Another characteristic that is basic for medical device regulation is the use of strong scientific evidence. It must be shown quantitatively and without any doubt that there is enough scientific evidence to show that this device is safe, and in the case of the US, if it's effective. You should also consider less burdensome means to testing. What this means is that you have to assure the FDA in this case, or at least also the notified bodies or the agencies responsible for medical device regulation everywhere in the world, that you not only have assessed the risks and benefits of your device, but that also you have done so in the least burdensome ways, and also within ethical scopes of testing of animals and of course of humans. That will include eventually the creation of a consenting form piece that must be signed by every patient that is used for testing, and also the submission of a protocol to an ethics committee and to the FDA in case that it is clinical. With this assured through an ethics committee and through a board that actually validates that you have consider least burdensome means, you can know that you have tested this for your endpoints in safety and or efficacy, but that you have also done that in the most human way possible. And finally, one of the basic principles of medical device regulation is to provide reasonable assurance of these endpoints, which is something particularly related to the design of medical protocols and clinical protocols for testing. But you should know in this point that the creation and calculation of an N, a number of patients required to create a statistically significant data, is the thing that is important in providing reasonable assurance of your endpoints. Finally, specifically for the FDA in its risk-based paradigm, the law gives the FDA this flexibility to calibrate the regulatory approach to the level of potential risk posed by new products. So you should know that even though many of the things that you can read on the FDA webpage or even on the webpage of the Regulatory Affairs Agency that is regulating your product in the nation of interest may have the capacity to change any of those requirements according to the possible risk that they see on your product. So you should notice that in order to be prepared for that, and also to try to design for products that might be easier to regulate or that might not pose potential risk for users. Okay, so if we move to device classification, we have talked that the FDA classifies devices according to risks. And you should know that a risk-based classification system is something that is pretty much harmonized already throughout the world. Nonetheless, even though we are going to see today the specific classification system of the US, you should know that this basic classification system is shared and that minimal, sometimes significant, but pretty much everywhere, there are minimal differences that must be met for you to recognize the specific way in which medical devices are regulated elsewhere. Okay, so in the classification of the US, what you should know is that it depends on the degree of control required to assure safety and efficacy of the device. And it is responsibility of the manufacturer or inventor at the moment of submission of their application to get regulating approval to regulate the medical device, to justify what is the applicable classification of the device in base of the judgment according to applicable law, and to the medical device classification that it's fairly harmonized throughout the world. So you will know that in the US specifically there are three main classes, class 1, 2, and 3, and the different classes are divided as risk is increasing. In this case, class 1 is the less or minimally risk classification, and class 3 is the one that is the most risky. Okay, so how does a class 1, 2, and 3 actually look like? Well, a class 1 in the US at least, it's basically a medical device that has minimal risk, it's definitely non-sterile, and it's of common production. What you're going to see here is that sometimes new medical products can be classified as a class 1 due to very low risk associated with it. But you can see that here in this slide, in class 1 there are many devices that are included that you can actually modify to get approval on a device that has clinically relevance, a significantly clinically relevant effect, but that also has a very unburdensome means to regulation, such as class 1. In this case, tongue depressors, casters, traditional stethoscopes, so the ones that are not necessarily electronical, wheelchairs, otoscopes, therapeutic oxygen tanks, inspection globes, even simple devices to correct hammer toe or calipers or to facilitate venous function or access can be included in this classification. For example, in this case, there is a new product called the Easy Vein, which is a simple class 1 device which actually had one of the fastest FDA approvals in history. For non-expedite devices. It actually took approximately 14 days from submission to approval, and it's basically the picture that you can see down there on the very bottom right, which is it's a device that inflates itself like a cuff and basically helps clinicians and nurses to detect the radial vein for traditional peripheral catheterization. As you may notice, this is a non-invasive, a minimum risk, a fairly simple device that has a clinically relevant impact, But it was classified as class 2 because of all these characteristics. So when you think about creating new medical devices, please try to take a look at solutions that actually are fairly known, and then try to see if you can come up with an idea that meets those requirements of non-invasiveness, minimum risk, and common manufacturing processes, so that you can enter into this space, which is fairly easy to enter. In the case of class 2 or other analog classification examples, as you're going to see, there are many devices that are classified in this cluster. I mentioned class 1 or analogous because in the case of Europe, there are two types of class 2 devices, class 2A and class 2B. We're going to explore those in a little bit more detail on further modules, but what you should know is that class 2 is a moderate risk or a classification in which, even though you may have devices that look fairly simple and unrisky, due to the uncertainty of materials used and how the manufacturing process is going to change those characteristics, the agencies sometimes choose to classify those as class 2 devices. In this classification, you have all the devices that are moderate risk associated devices, all the devices that have software or firmware, and many devices that have actually hardware on them. And of course, all the devices that have a purpose for diagnosis. And many devices you're going to see with a purpose for diagnosis are going to have software associated with it, and that's why they are classified as class 2. The idea behind class 2 classifications is that even though there is a moderate or minimal risk on it, the use of different materials or a little mistake or bug in software, or the idea that something might change because of a production process, gives a sense of uncertainty to the agency that makes them wonder if more controls should be associated to that device. And that's why those are classified as class 2. Within this group, you have digital glucometers, vital sign monitoring systems, antibacterial materials and devices created with antibacterial covering, such as these keyboards that I'm showing there. Any machines for in vitro diagnostic, non-invasive electrostimulation, pathological culture materials, electrical surgical instruments, other instruments to measure the parameters within the body that are non-invasive or non-implantable. Non-invasive meters for hemoglobin, for example, this new product from Massimo, it's actually able to measure a total hemoglobin concentration and hematocrit in a non-invasive way. So something that was actually considered impossible a couple of years back, and Massimo has already accomplished that through photonic effects. And finally, ultrasound and endoscopic machines, which all have either optics or diagnosis as the diagnosis purpose, which might be relevant for this classification in the sense that they have software or firmware or things that can change throughout processing materials. Finally, we have the class 3 devices. These devices are the ones that are commonly associated with a high risk benefit ratio. Most, if not all implantable devices are class 3 devices. An implantable device defines itself as any device, either passive or active, that is implanted in the body for more than 30 days. Devices that emit strong energy pulses, such as the gamma knives, are included in this classification due to the possible risk of failure of these devices. So examples of these devices are implantable pacemakers, cardioversors, breast implants, metal hips prosthetics, artificial ventricles, tricuspid valve replacement, artificial knee implants, aortic stents, or any other sort of stent, dissolvable or not. And of course gamma knife devices, as I have mentioned. Something really important to take from this is that device classification, as device classification grows, The amount of controls and of course of cost that is going to require for your clinical trials and controls in production are going to increase dramatically from class 1 to 3. It can be said that if medical device class 1 could be actually regulated by $100,000, a class 3 device is going to cost the manufacturer or the inventor more than $10 million to get into the market. So what you're going to see is that there is a abysmal amount of energy, time, and money that is going to be dedicated to regulate a class 3 medical device. And that could be a very important factor for you to consider in your biodesign process in terms of the money and time that you want to dedicate in order for this product, for your product, to be out in the market. In this sense, you should know that for a very effective dynamic biodesign process, you should try to go for devices that are either class 1 or at most class 2. Now that we know how classification of medical devices actually goes, we can take a look at some of the FDA specific review type processes that there are. As we're going to see, there are 4 different main ways in which a medical device can be either used in humans for either testing or regulatory approval for commercialization of it. And those pretty much encase all the possibilities that there are for an FDA review process. The first one is the investigational device exemption, the IDE, which is a process that allows you to investigate your device through a clinical study to collect safety and efficacy data. What you're going to see is that even before you can provide this into the market, you are most of the time for class 2 and 3, you're going to have to bring data on these 2 endpoints. And this is going to require you to do clinical studies on humans. And if it is an implantable or something in which there is substantial risk for doing this, you're going to require an investigational device exemption. And actually, for any device you're going to test on humans with these characteristics, you're going to have to apply for this permission. After having that, and after having gathered your data, either through an IDE or even if you didn't require to have this exemption for clinical studies, it's gonna go in which you're gonna have 2 different pathways to use in order to get regulatory approval for commercialization. The first is the pre-market notification, also called the 510K, and it's called the 510K because it comes essentially from the 510K section of the FDA law. It is a document that demonstrates all the different requirements that are needed for a class, for a device that either it has substantial equivalence or that is of minimal risk to get approved. What you're going to see is a pre-market notification or a 510K. It's used when it is possible to demonstrate something called substantial equivalence that we're going to define later on this module. To a legally marketed device, which is called, or that we're going to call a predicate device. And you should know also that multiple or split predicates might be used for this process to be justified. Nonetheless, you should know that split predicates are something not as used in the industry as it was years ago because of the agency's idea that split predicates might bring a higher degree of risk associated with newly commercialized products. Finally, the pre-market approval application for these 2 main regulatory pathways is a process used essentially for class 3 devices and some very rare class 2 devices in the US to evaluate safety and efficacy of all these devices. The PMA is essentially used for any device that cannot be proved to be substantially equivalent, okay, to a class 1 or class 2 predicate. And something really important to consider is that a PMA is substantially more costly than a 510K, not only in the submission part, but also in how your clinical are going to go on how your controls for manufacturing should go in order to prove that your device is safe and effective. A fairly consistent calculation that you can do is that a PMA is going to cost at least 10 times as a 510K approval process, which is something that you have to consider at the moment of designing your devices and getting into concepts of solutions, because of course you want to do your bio design process in the most cost efficient way. And by having a PMA submission on the line, it's going to be really likely that few investors are going to be willing to spend that much time and money on doing this without even consider that you could do or have the same effect by doing a more fairly easy regulatory approval process like the 510K. And finally, there is this humanitarian device exemption, which is a very particular review type process only designated for devices that are intended to benefit patients in conditions that affect fewer than 4,000 individuals, which was something that we mentioned earlier on this module. Okay, so what is now that you know what a 510K is, which is this fairly easy regulatory pathway for medical devices, you should know that it again stands under the idea of substantial equivalence and predicate devices. But what is a substantially equivalent device? Well, a substantially equivalent device is that device that, when compared with a predicate, has the same intended use and the same technological properties as the predicate. This is that a substantially equivalent device does not necessarily is identical to an existing device, but it definitely has the same intended purpose and the same technological properties. Okay, so for example, if you have a device that has characteristics A and B, you can try to search for a predicate that, even though has properties A, B, and C, it also has these two first properties that your device shares. And what is important is that substantial equivalence, to prove that, you have to be prepared to know all the technological properties of that predicate, and also to know what is the exact definition of its intended use, if it's inpatient, outpatient, home care. So for example, if you think about a pregnancy, a method to diagnose if you're pregnant or not, you can think about a test strip that can be done within the hospital or in a clinic with the supervision of a doctor, or those that are used on home and that you can buy off the counter. With this in consideration, the intended use of those three different devices are different because they are not used in the same setting, and that little change, that it's actually pretty big, can change dramatically the intended use of a single product, and of course its classification and definition, and that can actually hinder your capacity to claim substantial equivalence. Nonetheless, as we have mentioned, you can use devices that in combination have the same intended use and technological properties of yours to create a justification of substantial equivalence for your product. Knowing this, what are potential predicates for devices in the 510K process that you can choose? Well, you can try to explore devices marketed before 1976, because as we're going to see, the FDA regulatory process as defined now for medical devices was actually defined in that date. And so in 1976, every device that was marketed before that is not subject to a 510K or PMA regulatory approval process. And so you can use devices that were marketed before that. You can actually use two devices that were cleared as a 510K after that date. And you can also use devices that were exempt for a 510K process as predicate devices. Many people use one or more of these devices as predicates, as we have mentioned. But something that you should know is that a PMA also is going to be followed by another PMA. What this means is that you cannot take a PMA approved device as a predicate to define you as substantial equivalent to another device to be regulated to a 510K process. That may be done later on in future times where regulation changes, but by now all the different products that are class 3 are subject, all of them, to the classification, the class 3 classification, and of course to the PMA process, which we have defined as the most lengthy and costly of the ones that we have already explored. And with this in mind, please try to take a look at our biodesign book on its chapter 4.2 on regulatory basics to know more about the FDA regulatory approval process and all the different things that you have to meet in order to get your device regulated. And with this, we have concluded our regulatory basics chapter on our biodesign course. Thank you. My name is Luis Swanksen, and see you later.